Okay, continuing our discussion about enzymes. The last lecture we went through some basics about what is an enzyme and how do they work. We're going to talk more today about more specifically how they work. Now, this slide here, I'm trying to explain here and talk about how the enzyme and substrate interact. Now, we've been saying just loosely here that the enzyme binds to the substrate and causes a change. Well, here's how the change works. So I put up here. Remember, there's all kinds of little bonds holding together these molecules. Okay, and the substrate and the enzyme bind by very weak, as I put up there, ionic bonds, hydrogen bonds. Any kind of bond like this is very weak, and the enzyme causes a change. Kind of like puts a stress on the substrate, like stretching those bonds, and it may cause those bonds to break, in which case now you're going to get two substrates, or two products, excuse me. Or it may cause attraction between the two and actually join something together. Now, the breaking apart, catalysis here, it begins when the amino acids that are making up the enzyme. Remember, those amino acids have little R groups, little sidebars coming off that give the protein its unique shape and characteristics. Those R groups are usually going to be the things that are going to cause the splitting or the combining of different substances, different substrates into products. Now, enzymes can catalyze thousands of reactions every second. We go through when we draw on the board, of course, it goes really slow, but these reactions in real time go extremely fast. Now, it would stand to follow then that the amount of enzyme and substrate are critical. The enzyme rate or the decomposition rate will always increase until there's a saturation point. And we'll look at this really close when we start dealing with photosynthesis, especially. So long as there's a whole lot of the substrate available, the enzyme action will steadily increase until it hits a plateau. When the enzyme activity levels off or plateaus, then you know that there's not the substrate. The amount of substrate has maxed out. There's not enough substrate. There's not enough enzyme to handle the substrate available, and we'll see that a lot with we study photosynthesis. Dealing with enzyme activity, we talked in class and lab here. Most enzymes in our body are going to operate around 37 degrees Celsius. If you guys had my anatomy class last year, those of you that had anatomy, a lot of the digestion labs we did, we had you guys make water baths set at 37 degrees Celsius so we could simulate the action of the enzyme. But this being AP Bio, you have to understand, there are some organisms that strive at much higher temperatures. Thermophiles, so thermophilic organisms. We have some bacteria that strive in hot water geysers. They survive at these extremely high temperatures. So what we're showing with this graph right here is this. An enzyme, if you're looking at the temperature across the x-axis, on one particular enzyme for a human, it slowly increases its speed, reacts faster and faster until it gets around 37 degrees Celsius, normal body temperature. At that point, beyond that, the enzyme is denatured and the activity slowly decreases. Whereas some thermophilic organisms living in those hot springs or underwater vents, they don't even start working until you get around 40 degrees Celsius. Then they slowly increase until you get around 70 or 80. Higher than that, the temperature drops off. Which has a huge influence on enzymes. And again, in humans, most enzymes are going to operate around 7. But some enzymes, for example, pepsin, those of you that remember the anatomy, pepsin is a digestive enzyme for breaking down proteins that you find in the stomach. Pepsin works best in a pH of around 2 to 3. So if you were to look at the graph again, most enzymes are going to be operating somewhere around in here. If you're looking at it, most enzymes are going to have a graph going up, probably peaking right around 7 and then back down. But in humans, in the digestion, here, pepsin works best in an acid environment, like in the stomach, where trypsin works best in an alkaline environment. Trypsin is found in the small intestine, where you do have an alkaline environment. So even digesting proteins in your bodies, the enzymes control it. So they include most of our vitamins. Pause this. Okay, so again, things that are going to affect the enzymes. The last couple slides we talked about the temperature, the pH, the presence or absence of cofactors and coenzymes. But provided all that stuff is there, enzyme activity is still influenced by other factors. Sometimes an enzyme's activity can be affected by the presence of what's called a competitive inhibitor. As you would guess, the inhibitor is something that stops the enzyme's action. Now remember, from my pictures here, here's the enzyme, and there's an active site on this enzyme that has all the necessary R groups for the amino acids, and it's going to form a particular shape that the substrate will fit into. A competitive inhibitor is simply some other substance that will bind to the enzyme because one part of the shape is kind of like the shape, matching the shape of the enzyme. When this competitive inhibitor gets in here, it stops the substrate from actually binding to it. So you can add as much substrate as you like, and it's never going to bind up, and you'll never have the breakdown. So if, for example, we did have a competitive inhibitor for catalase, it would bind to the catalase and would stop the hydrogen peroxide from binding to it, and therefore we would never split the hydrogen peroxide. But if you have a competitive inhibitor, you can also have a non-competitive inhibitor. Non-competitive just simply means they are not competing for that same site. So what happens is the non-competitive inhibitor, that are showing right in here, the non-competitive inhibitor binds to another site on the enzyme. And when it does, it changes the shape of the active site for this enzyme in such a way that the substrate can no longer bind to it. Now, unlike competitive inhibition, non-competitive inhibition is reversible. So in non-competitive inhibition, that non-competitive inhibitor can be kicked off, and now you can actually start binding the enzyme to the substrate and catalyzing the reaction. So in this case, adding more substrate will actually start causing the reaction. With competitive, you can add as much as you want, nothing's ever going to change. Non-competitive, you can add more and more substrate, and you can eventually start getting a reaction to proceed. Now sometimes it's not the presence or absence of competitive and non-competitive inhibitors. Enzymes do have to their shape. We call that allosteric regulation. Enzymes can be found in the bodies in active forms and inactive forms. In my picture right here, for an enzyme to be active, it may have to have a particular shape, like this. But when the enzyme is not being used or triggered, it goes back to this inactive form. So the reactions aren't always being catalyzed. Now, this thing will switch between the active and inactive forms. In allosteric regulation, some substance that we call the activator binds to the allosteric site, like right here. When it binds to this site, it prevents the shifting back to the inactive form. Now the enzyme stays open the whole time, and the substrate is continually being broken down. You could also have an inhibitor. If you look at the shape on this side right here, the allosteric inhibitor will bind here and keep the inactive form from shifting back to the active form. So the enzyme stays neutralized. This is what controls all the enzymes making up your cells. In particular, it's easy to think about this when you think about sports and the applications in energy systems of sports.